In this video, we will focus on the second class of topoisomerase called Topoisomerase II. These enzymes change the linking number by cleaving and religating both strands of the DNA double helix, and they can separate interlocked double-stranded DNA molecules. Topoisomerase II cuts both strands of the DNA helix, passes another segment of the double helix through the break, and then reseals the cut. The linking number changes by 2 during each round of catalysis because the enzyme manages 2 DNA crossings at once, thus changing the rise without altering the twist. This mechanism ensures that DNA supercoiling can be precisely regulated. Topoisomerase II also plays a crucial role in the separation of interlinked circular DNA molecules, a process called decatenation. During decatenation, the enzyme cuts one of the DNA molecules, which allows the other to pass through the break and effectively disentangles them. The opposite process, where 2 circular DNA molecules become interlinked, is called catenation. Topoisomerase II can both catenate and decatenate DNA. Here we have a table that illustrates various features of topoisomerase II. The active form of the topoisomerase is always multimeric, comprising several subunits. In prokaryotes, it forms a tetramer, while in eukaryotes, it forms a homodimer. The cleavage of both strands of the DNA double helix leads to the formation of 2 phosphotyrosine bonds. ATP is necessary for the process of topoisomerization. The DNA can either be decatenated or catenated by the topoisomerase. In prokaryotes, class II topoisomerase can convert positive supercoils into negative ones, while in eukaryotes, it relaxes both positive and negative supercoils. The topoisomerase's mechanism of action is the double clamp model. The structure of the topoisomerase enzyme in prokaryotes is heterotetrameric. It consists of 2 subunits, A and B. Each subunit is present in 2 copies, which forms a natural tetramer. Yeast topoisomerase II, or TOP2, helps untangle DNA by cutting both strands, passing another segment through, and then resealing the break. This process relieves twists and knots in the DNA during replication and cell division. The topoisomerase II enzyme in eukaryotes also comprises two subunits. Each subunit is composed of two domains, which are analogous to the A and B subunits in prokaryotes. When combined, these subunits form a homodimer, which is the active form of the enzyme. These two domains and subunits are connected at the top and bottom of the structure. Now let's look at the mode of action. Once the topoisomerase II is bound to a double-stranded gDNA, it utilizes its two catalytic tyrosines positioned on each side of the DNA molecule. The hydroxyl group of each tyrosine cleaves both strands of the DNA. However, this cleavage doesn't occur at the same position on both strands. Instead, the cuts are staggered, which affects two different phosphodiester bonds at different points along the DNA. As a result, two phosphotyrosine bonds form, one on each strand. Subsequent conformational changes in the enzyme move the cleaved strands apart, which creates a gap between them. ATP hydrolysis is responsible for these conformational changes in topoisomerase II. Through this gap, an intact double-stranded tDNA molecule can pass. Further conformational changes bring the two strands close together again, which reinstates the two phosphodiester linkages in the gDNA. This process sets the stage for a new cycle of topoisomerization to occur. We commonly describe the two double-stranded DNA fragments implicated in the topoisomerization as the G and T segments. The two strands of the G segment are cleaved by topoisomerase II. The T segment is an intact segment that passes through the gap formed after the G segment cleavage. In the first step, the topoisomerase opens from the top part, which separates and moves the two domains, B and B prime, apart from each other. The topoisomerase acts as a clamp with the top part open, which allows the G segment to enter the catalytic site that the B and B prime domains define. ATP then binds to the topoisomerase and triggers a conformational change. This allows the T segment to start entering the upper clamp. Next, phosphotyrosine bonds form, which cleaves the G segment. These bonds are between the A or A prime domain and the G segment. Another conformational change separates the two ends of the cleaved G segment, which allows the T segment to move across the gate and be trapped in the cavity of the closed topoisomerase, which has a diameter that is large enough to accommodate the double-stranded DNA. Another conformational change then opens the lower part of the topoisomerase and releases the double-stranded T segment. Finally, the G segment is religated, ATP is released, and the cycle can continue. At the end of the topoisomerization process, instead of adding a new cycle of topoisomerization, the upper clamp opens again and releases the G segment. Gyrase is a type II topoisomerase that is unique to bacteria. It can convert positive supercoils into negative ones and prevent the formation of positive supercoils, thus maintaining the degree of negative supercoiling in bacterial DNA. Gyrase has a higher affinity for relaxed DNA than for negatively supercoiled DNA, which allows it to ultimately maintain the degree of negative supercoiling. Bacterial gyrase also contributes to the compaction of DNA in bacteria. The negative supercoils added by gyrase store energy, which is not lost. You can think of it as the energy that is stored in a compressed spring. The release of this energy facilitates the separation of the two strands of DNA during replication. DNA gyrase interacts extensively with DNA, with 104 base pairs of DNA wrapped around one molecule of gyrase. Eukaryotic topoisomerase II and bacterial topoisomerase IV, unlike gyrase, cannot introduce supercoils. They can only relax both positive and negative supercoils. In eukaryotes, the introduction of negative supercoiling favors DNA replication. However, it is not catalyzed by topoisomerases. Instead, it occurs when DNA wraps around histones to form nucleosomes. In conclusion, class II topoisomerases catalyze the topoisomerization of DNA by cleaving both strands of a DNA molecule to create a gap and promote the passage of intact double-stranded DNA through this opening. They can change the supercoiling if both the cleaved and intact double-stranded DNA belong to the same DNA molecule. Alternatively, they can decatenate or catenate DNA by separating two double strands from the DNA molecule if the cleaved DNA and the intact DNA belong to distinct DNA molecules.